So I'm going to thank the four speakers. And we have about 45 minutes now to begin to open some dialogue. And I got a hand up in the front row. So we'll open for some questions, and then I want to follow up with some redirects, if you will, to our panelists.
Again, this is Cheryl Walker, a member of the committee. This has just been absolutely fascinating, and I think for toxicity testing and validation, this is absolutely the right place to start. It seems to me, though, that there is an incredible opportunity to be able to use these models in the setting of prevention. So prevention studies are very, very hard. They require large numbers of people. They're longitudinal. And so I would wonder if any of the speakers have given some thought to modeling pathologies in these systems, and if so, whether that could in fact give us opportunities to look at agents that might prevent environmental diseases.
That's a question to the audience.
Cheryl, that's an excellent question. This is Meho, Cincinnati. That's an excellent question, because most of these systems, as I'm seeing them, they are kind of like normal tissues. So they're looking at the drug response. So mostly at pharmacological agencies. Now, the questions I actually have are actually with dosimetry, toxicity. Many of the toxicants does not have a linear relationship, whereas when we look for drug responses, we're looking for a linear relationship oftentimes, or we're looking for a threshold where we're beginning to see responses. So how do we reconcile these differences where in toxicology studies we may not get a linear responses versus some of these systems that you have designed, mostly focusing, or maybe primarily focusing on looking for that threshold dose?
Okay, I don't have an answer for that. Yeah, we're 0 for 2 with the questions. But I think it all goes down. When I first started this type of work on myself, I mean, I've always come from a no AL type of point of view, and those doses. It's always been from the very beginning. Whenever I've started with animal work, and then trying to do IV, IVC, the calculations that it's not linear, first of all, on how the dose response happens, and then it's not the same for what the equations that the community uses to back calculate what the right dose is for the in vitro system, be it monolayers or co-cultures or in fluid dynamics, or for what organ you're talking about. So the questions are right there, and I think that's what this group needs to talk about, but also bring in the modelers, the mathematicians that have worked on the equations, and then bring this into it as well. You're an expert in this too.
So if I can ask you to hold for just a minute, there was a hand up over here with a mic in hand, so we're going to go to Yvonne first.
I just wanted to tell you I was going to give you an answer to the first question. So
all right,
I'm okay with waiting.
Answer the first question then.
So
thanks.
The question was about pathology, incorporating pathology into these models. So I was waiting for some of the performers to jump up, but they're too reticent. So as an NIH person that's part of the Microphysiological Systems Review group, I can tell you that the DARPA solicitation originally had a lot of interest in trying to develop systems that would also do pathology. Specifically, they were very interested in things like vaccine development and the opportunity to use these systems to try and do vaccinology, which of course streaks directly to prevention. And in the NIH arena, we were highly encouraging people in the solicitation phase to try and look at normal physiology, but also to mimic some aspects of pathology. Now, you have to walk before you can run, and so most people are really very focused right now on developing a lot of normal tissue because they have to be able to compare this so-called pathology to something, right? But I can assure you, as one of the people who looks at the progress reports, that it is very much in the minds of the people that, at least that we are funding, who are developing these systems to look at long-term and even short-term incorporating pathology as they can. And that's one of the reasons they're very, very excited about using iPS cell technology to generate tissue types, because there is a really early opportunity to try and incorporate things that have at least a genetic basis of disease. So I hope that's a partial answer for you, and thank you.
Could you just state your name for the folks on the webcast?
Yes, I'm sorry. It's Rosemary Hunziker from NIH.
And just a minor housekeeping matter. Since we've jumped right into the science, which is what we were going to do after the break, and Drs. Bahinsky and Kolajay were going to join us, might I ask the two of you to join us now? And I'll So Yvonne, and then back to the mic.
So, Yvonne Drusen, I'm a member of the committee, University of North Carolina. So I wanted to come back to the topic of this discussion, which is scientific challenges as far as application of this for environmental health is concerned. And my question is to Roger. I mean, we actually did invite you to be a paleontologist of sorts, because it seems, at least to me, that many things in science and in application are cyclical in nature. So the struggles or the concerns that you were expressing about some of these early, more complex constructs such as skin and eye, right now, at least in Europe, the regulatory pressure is not to use animals at all, but to use some of these models as it. And so we're not talking about having human data. We're not talking about even having in vivo animal data. So what has been your experience as a person kind of, in the middle between the manufacturers and the regulators of, thinking of what are the scientific issues that you have to help the manufacturers to solve in order for the product of your work to be accepted by regulators in the context of the changing or shifting regulatory requirements?
How much time do we have? Good question. We do act as an interface, and many laboratories act as an interface between industry and regulators. But you also have to work one-on-one with both areas. So with industry just itself, you have to be able to convince them that the model that you're using and the approach to using that model makes sense, is reflective somehow of what is going to happen in the real world, that the type of exposure that you're using is related to the exposure that would occur there, and that subsequent applications are too. But you have to be able to describe the system, and I think that's very important as you're talking to the regulators. You can't just say it's a model of X. You have to be able to have some descriptors there that are meaningful, that are physiologically relevant, and that are provable, I guess, each time. If you're providing a report either to industry or to the regulators, there have to be numbers, data in there that qualify that tissue and qualify the use of that tissue for the purpose that you're putting it. The data can't just be the number that came out when you put chemical X in there. There has to be, more than that. It's, in many cases, it should be a historic reference. It should be over the last 3 years with this tissue, values for using material X has been 25 plus or minus 2 standard deviations or something like that, so that there's a real quantification of how your tissue is reacting this time to how it was before. And those, I think, things are looked at very carefully by the regulators. You have the whole good laboratory practices question. And this becomes important, again, in having knowledge of the system, because under GLPs, there's a study director associated with the study, and that study director takes immense responsibility in signing a report because he or she, at the end of the day, is then responsible for those data. And there are certainly legal ramifications, there are fines, there are jail sentences for not really knowing what was in that report and not being able to support that at some later time. In fact, the FDA right now is really, at least in the last inspection that we had, is encouraging people to have site visitor to, do on-site inspections of their reagent manufacturers. That's just probably over the last 2 or 3 years has started to become important. So that can mean, and we do that for our tissue manufacturers now. We're probably one of the few who do. We actually go into the tissue manufacturer. We look at their quality certificates, their quality procedures. We look at their controls for the various batches of tissue, and we look to see whether they have a quality control program that we can accept. Otherwise, as I said, a study director is signing a report using something that he or she knows nothing about, and that's a pretty risky thing to do. I think I've lost track of the original question right now, but I'll say.
Next, please.
Jonathan Himmelfarb, University of Washington. I was going to be reticent, but maybe a little less reticent than some of the others in responding to Cheryl's question, with some comments that I think reflect what Rosemary was saying. At least from my understanding of the NCAT's view of this entire program, is that which was predominantly focused on drug development, is that the drug development process is broken really for two different reasons. One is safety, but the other is efficacy. So most of the discussion today has been on the safety side, and the equivalent in drug development clinical trial failures are the off-target toxicities that were unexpected. But the flip side is probably equally important, and that is that there are many drugs that are very effective in animal models and given disease states that turn out in clinical trials not to be efficacious. And the hope is that these systems, can be used to develop disease models in addition to simply looking for toxicities that can be looked at for efficacy, whether it's a drug or it's a preventative approach to an environmental toxin. So example I'll just use in our system. One of the reasons that we decided to include perivascular cells or pericytes in our system is that recent data in the kidney suggests, mostly from lineage tracing studies in animal models, that the predominant cell that transitions into a myofibroblast that causes fibrosis in almost every chronic kidney disease is the perivascular cell, which when the vessel is injured detaches, moves into the interstitium, undergoes a transformation and contributes to scarring and fibrosis. So we, from the beginning, have thought of our system as potentially capable of being a disease model for fibrosis in the kidney. Now, as Rosemary said, you have to walk before you can run. We have to get our system up and running and optimized first. But that would be one example of how these systems could also be used to look for preventative approaches.
Can I just ask a quick follow-up question?
Yeah.
Are you all looking at Cheryl, we have so many people on the web that want to hear you.
Remains to be seen. So Jonathan, are you all looking for, by any chance, it seems your model is already poised to look for cyst formation, for instance, for polycystic kidney disease defects and so forth?
Yeah, so that's certainly of interest, and that's where, for example, there are very recent papers published using iPSCs in the last couple months from individuals with polycystic kidney disease and showing you can develop a phenotype not in a 3D system, but simply growing cells in a 2D system. So if we can move iPSCs towards renal tubular epithelial cell lineages, that's an example of where drug discovery could take place in this kind of a system. And we have thought about that.
So I just wanted to, following up on that, so you've opened up Pandora's box on disease models now. So a couple of the tissues that I didn't show today that we are looking at specific disease models. So Kit Parker's group put out a very nice paper just recently in PNAS on Bart syndrome, which is basically a genetic disease of Bart's, B-A-R-T-H. It's a mitochondrial defect, and it's a single genetic mutation. And so they were able to develop it two ways. One is using Kyle's technique of IPS, so taking IPS patients from the IPS cells and developing those into cardiomyocytes and recapitulating that disease very nicely, the structural defects that occur. And then second was, again, something that Kyle mentioned, using CRISPR to go in and edit that gene in normal cells and then again recapitulating the disease. And by doing that, they were actually found a pathway that was novel, reactive oxygen species, and actually gave a scavenger. And so there was a novel pathway that they could explore for a therapeutic. So I think, as Rosemary, rightly pointed out, I think the genetic diseases, especially the single gene mutation diseases, might be easier to study, at least initially, in these types of systems. We can either recapitulate those using IPS cells from diseased patients, or going in and actually introducing, using CRISPR or other gene editing techniques. Where it's going to be a little more difficult is kind of these long, progressive, chronic, multifactorial diseases and recapitulating those. We may have some success with those, we may not. But we are, moving towards, doing those types of things also.
And worth mentioning, then you could study environmental influences that might promote or exacerbate the development of those diseases as well in these models.
Exactly. And so another model that we're developing, it's fairly far along, is a small airway model. And so we're actually starting initially populating those cells with normal, bronchial tracheal cells. And now we're actually looking at COPD patient cells as well as asthma, and then looking to see things that might exacerbate the condition or not. And so we're thinking about those and developing those models. And going along, with the regulatory guidelines, I think, Roger's comments are actually right on the mark. I think industry will adopt those, as Jonathan said earlier, because there's a gap there for efficacy. And I think the regulatory hurdle isn't there. So they're really being used to make business decisions within the companies. And so the bar is a little bit lower. Once you move into the regulatory arena, I think things like context of use and fit for purpose are going to be very important, because you're only going to use those one model at a time. So in insert year, I think you're going to reduce the animal usage one model at a time, and hopefully over the course of time, take bulk of those out. So.
Since I was able to sit there all the way till question one, until I got brought up here, I thought I'd give my answer to question one as well. So from an IPS field perspective, and it's about a year and a half now since I've seen a good review of sort of a mapping of all the different diseases that have been made into IPS cells. But it's in that paper is about 50 different diseases, and these are ranging from sort of the simple ones that Tony mentions in terms of single point mutation that have a very clear sequela like long QT syndrome and all the various flavors of that. Ranging to things like clastogenic events like Down syndrome, but also even more complicated polygenetic diseases like diabetes, where by and large, even in the sort of monolayer, simple single cell systems, cell type systems, people are showing a clear phenotype of disease. So my feeling is, as we get better at really two things, the microphysiological systems and getting additional cell types in there, so adding astrocytes to neurons, for example, or Kupffer cells to hepatocyte models, and then integrating some of the things that Lance talked about, which is much better detection systems of identifying events that aren't just dead cells. One of the things that always struck me when I was at Roche and we were doing all this work is that they would spend all this energy making cardiomyocytes, and then we'd say, okay, here's our IC50 curve, sort of just smash this very expensive toy that they just gave us. And so when we got to the point where we're looking at and asking better questions, so looking at subtle effects, whether it's changes in arrhythmia that I showed, or looking at neurite outgrowth, or subtle injury markers that aren't quite death, that helps a lot in terms of understanding, the potential flexibility, response of the model, et cetera. So I look at it as really, all these things will come together into these microphysiological systems, and the underlying data are already there showing that it's headed in the right direction.
Okay, moving back to the microphone.
Thanks. Rick Becker, American Chemistry Council. Just an observation, and then I guess a question probably, but it seems to me, and this kind of follows up with what Rusty said, which is, and the theme of fit for purpose here. one of the areas we've grappled with kind of, and I think everybody does right now, is we can kind of understand what happens at the molecular level. You know, the interaction I'm talking about, let's say, adverse outcome pathways, which is a linkage of key events that start with the interactions that, you know, molecular levels proceed through cellular responses into organ responses in the whole animal. It's a very useful construct. It's a little too linear right now, right? But it helps organize your thinking about how one could proceed to develop a screening and evaluation approach using some of these high throughput techniques or lower throughput, high content techniques. But I always run in, I've been working in this area for the last year or year and a half, I run into this blockade, if you would, right when I get to the organ effects, right? Because then I'm right back to the whole animal systems. Because that's typically where we have the data. And so if I want to probe that, if I get to that point and want to probe it, I don't have the tools just yet. So I think these are going to be excellent tools that will allow us in AOPs kind of to get to that level and then probe it from a dose response perspective, probe it from if you would, a pathogenesis perspective, as we've heard about today in terms of the actual changes that one would see at a physiological basis in terms of dose response, the kind of the tipping point kind of questions that we always get. So I see a lot of benefit in this, and perhaps sooner rather than later, because we do run into this thing with animal models. And so if we can kind of begin to look at some of these that are more common, I think of the common things we run into with cytoproliferation as a promoter for liver cancer in animals. If we could understand that through a liver model, that might help us make those hurdles a lot quicker and not have to do chronic animal studies just to show that we get cytoproliferation that leads to liver tumors in animals. So that's kind of a comment. So I want to kind of encourage folks to think about these kinds of tools in the regulatory framework as kind of that fit for purpose. Take us back to where we can say, how can we link these things together to make a whole, but in a decision that will help us move not to having to do all the testing that we normally have to do just to prove something that we probably already suspect. So I guess that was a comment. Thank you.
I
guess I'm going to follow on the comment theme.
Could you identify yourself, please?
Maureen Bunger. I'm Psychon Innovation. So I've had the opportunity and pleasure to work with a number of small businesses creating tools to support the science in this space. And I wanted to connect together what Roger was saying about the need of the commercial domain to get involved to produce the more high quality, validated qualified systems, and to tie that with what Kyle mentioned is that CDI, if you look at them as an example, has spent $35 million qualifying and quality controlling the tools. And so I fear that a number of these model systems that we're looking at today are simply going to fall by the wayside because there's not the financial support to qualify them and validate them. And I would be curious to know if there have been thoughts on how to overcome that particular barrier.
All right, that's a great question about how we get them, if you will, into practical use through some kind of validation. Anyone want to take that one on?
So I guess I can try to address it. To me, it's one of these things where the economics of the outcome has to drive the need for it. So, and the arrhythmia story that I told, that's a much bigger group of individuals. But there's clearly a need in the pharmaceutical industry to do a better job of identifying cardiovascular risk earlier on. And so that need was so great that the FDA recognized it. And so that pushed a body like HESI to bring everybody together, the right groups of people, to drive that validation. And funding becomes secondary to the sort of the greater scientific good of accomplishment. So I think, and I also use that number more from the $35 million, more from a quality management enterprise-wide system for manufacturing of everything that we've done to date. So it's more of a McDonald's type number of what it has cost for us to be able to do this. But in terms of individual assays, it's not that large at all, I don't think. It's really a matter of making sure that you have inter and intra-lab variability control, that you have you have a protocol that's robust and reproducible, that has an output that can be used to make good decisions consistently. So that it wouldn't cost that much, I think. But what drives it is scientific need and then economic need ultimately.
Yeah, and just to kind of follow up on that, I think a part of it is on the the philosophy of the institutions that are developing too, and I think they have to have that mindset early on that this is not an academic exercise. There really is a commercial potential down the road, and the V's can be used as an example for that, because one of the tenets of our becoming a research institute for high-risk research is also commercialization and translation, and so we're always thinking about that down the road. How is the instrument going to be designed? Is it going to be user-friendly? Can we make these chips? One of the first things, and it's been highlighted a couple times already this morning, is the material, PDMS. I mean, it's great for prototyping. It's got a lot of issues as far as manufacturability and absorption of small hydrophobic molecules. So that's not going to be the commercial product. And so early on, coming from a pharmaceutical background, we saw that right away, and so we partnered with Sony. And so we have looking at different materials to try and go around. So we have internal efforts to look at alternate materials, as well as external collaborations with companies like Sony, but also manufacturability. So looking down the road, right now we're making hundreds of them, but we want to have the capability, if these instruments actually want to be useful and out there in the workplace, to make thousands, hundreds of thousands, or millions of these chips. And so I think you need to have those early partnerships early on with these companies and actually thinking about it very early on in the development of these technologies. Otherwise, you're going to be at the end stage with a cool technology that isn't usable by the general public.
Yes, Yvonne.
Just to follow up on this, I think a lot of the discussion and the costs and and why these get on the market is driven by the pharmaceutical applications. And here, while pharmaceutical is important, this particular meeting is about environmental health, and the regulatory pressures and the needs for manufacturers to test certain things is absolutely not on the same level, because the regulatory pressures are completely different. Therefore, I was interested more in terms of, again, the European experience, where, the European Parliament just decided that it ain't going to be like this anymore, and then you either do it or you perish, rather than it's more of an evolutionary period of time. So I think, can any one of you comment on, how is this going to go down for, more complex, wider chemistry, really uncertain regulatory regime, because you really don't know whether you're shooting for LD50 replacement, a 90-day replacement, or 2-year cancer study replacement, because everything kind of matters, and you never will have the human study. So it's a little more complex, I think. So I was wondering if we can maybe have that discussion about the drivers of developing this technology and putting it into the users, that they will be quite different and maybe much weaker in the environmental health.
I think we'd probably all agree with you. The difference I see, the somewhat difference I see in environmental health, is the number of materials that may need to be screened. Even though there are large libraries within the pharmaceutical groups, and that leads to a large amount of testing, I think still most people, with knowing that there are regulations within Europe and within the US and even within China now that require large-scale screening of millions of compounds, that's a driver that's within the environmental health perspective. And whether it's done by utilizing things like tissue on a chip, or with the larger scale screening programs that are going on with less sophisticated systems, and moving from one to the other. I mean, you're involved mostly in prioritization with some of the existing screening programs that go on now. So you could see this being a necessary secondary, tertiary level, at least in my mind.
So I'd actually like to make a follow-up to that with and a slight modification in the direction, because I want to remind you that tomorrow we're going to beat the regulatory issues here relative to these tools. And so I'd kind of like to save some of that for tomorrow and focus us today, if we can, more on the science of where these tools are. But if I may be so bold, I would like to ask those of you who are actively engaged in this, if Yvonne Drusen walked in and said, we'd like to use this to screen 100 chemicals, do you believe your technology is ready today to do that? Or what would you project for a practical use? And I'm just putting that out there, and then we'll kind of move back to the science. But where is the state of the science today to be able to use this in a routine basis? Are we a month away, 10 years away, a lifetime away? I honestly would ask Lansing, John, right? Sorry, and Tony and others to comment on that. So I see, yes, please.
So, should I jump the line? Yes. Sorry.
Yes. Sorry. There are no rules in this game, as you can tell.
Apparently not. Lance Taylor, University of Pittsburgh. So I would characterize the state of the art today that in our hands, with people that have learned how to nudge things through these advanced but still prototype systems, we do 10s to 20s of compounds and can generate data that's reasonable. We deal with the issue of binding of some of the hydrophobic small molecules to the devices by looking at the inflow and the outflow by mass spec. So it's not high throughput, but I would contend that it's relatively high content from the standpoint of the kind of information. So the kind of things we're doing today are more collaborative. In terms of when do we think just a simple liver device, because that's one of the concepts we're working on. You can have a liver alone, a kidney alone, or start linking 2 or 3 together as an early stage. I think it's within a year. I think it's safe where the organ systems alone will be robust enough and reproducible enough. But it's going to take that much time. And I would not recommend to people that we kind of send out devices and cells and have them start using it alone. We're going to start doing linkage and across a couple of laboratories within the next few months. So I'd say about a year. Otherwise, it's good collaborative effort.
But just to clarify, I think that the throughput is probably less important because, I mean, most of the reason why we need, or or at least particularly my center is using higher throughput is the legacy issue, right? We have a lot of we have a lot of chemicals we haven't tested, and just because we haven't tested them for the last number of years, right? We've kind of backlogged them, right? And so we need to get some throughput to get through them. But going forward within most of the chemical industries that I know of, and that particular application, whether it's cosmetics or whether it's more commodity chemicals or even specialty chemicals, they don't have a large number of chemicals they test for a particular application at any one time. And so if you're bringing up a particular system for screening cosmetics in Europe, or whether it's another particular chemical for a particular electronic application, right? It's not like they have a whole range of 100 molecules that they're going to narrow it down to pick the one, right? It's probably less than a dozen and probably around a half a dozen that you can screen in that particular context. And so it's not really a throughput issue at this point.
Yeah, I would agree with that. I think the way, if I need to restate the question, is the science. When we think about, I hate to refer to this simplistically as methods development, but what is the time frame by which we believe these systems will be sufficiently validated, for lack of a better word, or debugged, that they can be used as scientific tools? Is that now? I heard about a year as one example or one answer.
Jonathan Himmelfarb, University of Washington. So I think a lot of my comments would echo Lanza's comments. I think of these as high content, high value systems, certainly not high throughput systems. Yep. But nonetheless, they can be used as they develop to test moderate numbers of compounds. Our work plan for the year coming up is to test 12 compounds that are known nephrotoxins in the system and see if we can generate molecular signatures for the types of toxicity in the system. So we'll use a family of aminoglycosides as an example. I showed you some data on cyclosporin. We'll look at tacrolimus and other calcineurin antagonists in the same system. And then hopefully we can get different patterns of toxicity that could be replicated for testing unknowns. I think the problem would be it's the sort of known unknowns versus unknown unknowns. We would have real trouble testing a chemical that had some toxicity that wasn't in one of the patterns of previous nephrotoxins that have been identified or tested in our system. But our work plan is to test 12 compounds in the coming year and a family of 50 to 100 compounds in the 3 years, over the 3-year period of time. So I think within some range of 1 to 3 years, I think some of these systems will be robust enough to do some of this kind of testing.
Yeah, I think I'd agree with that also, and it's very organ specific, right? And so some of these are much more advanced than others. I think the liver and the kidney and the lung are probably fairly far advanced that we could do that. Again, with the caveat that Lance said is that these are very engineering intensive assays at this point, and so I think they need to be done collaboratively or within spin-out companies that may evolve out of these types of technologies over the next couple of years. I don't think they're ready for prime time to be sent out to laboratories or individuals to set them up on their own. So yeah.
And if I can be so bold, just to stay on this, I got a hand signal, and I think Julie is going to try to answer as well. So Julie and Bill, and then we'll come back to you. Sorry.
So if I could throw a wrench into the system and say that if we add the female hormones, it could be maybe a little longer, or stated another way, we are ready to give you the hormones, the follicles, to test in your systems. They are producing what they're supposed to be producing. The only caveat is that we need to test your media to make sure that the follicles are happy with the media, etc. But I think it's an important layer that we need to include, and that yes, it could maybe delay things in terms of validation and confirmation. But I think in terms of that tool, as we mentioned, fit for purpose, to provide the hormones, I think we should be testing them very soon.
Okay, and then we're going to go to Bill, and then back to you. I'm sorry.
Okay, I'm Meho. I'm tired of waiting. Okay, so I'm also a member of the committee, but the microphone is always on this left-hand side. So
we have microphone hogs.
So I really want to follow up a very, very really technical question. So most of your cell lines actually have been living in a very controlled environment. So it's already asynchronized from a circadian rhythm of human. So that's point number one. So how realistic it is to be able to test for toxicity. Second question is a question. None of mine are comments. Second question is about disparity. Genetical disparity means different groups. Racial, ethnic groups, that's important, because even in the pharmaceutical company, you already know that if you give the same dose to a person in Asia, they will die, they will faint, because of either the background or their body sizes or the metabolism are different. So how would we use this kind of system to actually broaden it to perhaps, for example, 1 million out of 6 million of Chinese, okay? So that's a major issue. The third point I am actually going to ask is when you are dealing with iPS cells, I would have to say, with a very limited knowledge, I understand that it's a major reprogramming of epigenetic reprogramming. That means a lot of the past histories are probably not being registered and and being recorded. So are these systems useful for us to look at toxicity in the future that is related to perhaps being able to apply to humans that have lived through many, many years and multiple environments? So this kind of environmental and toxicity interaction would be able to address. So, but this is too complex. It's just like 3 tough questions. Maybe, hopefully, we can get a small bit of answers.
I guess I can try to answer the third one because there's no answer. But I mean, you're going to need. I mean, the field's relatively new, obviously, and so a study that would have to be done would be making iPS cell lines from an individual over their lifetime, and multiple individuals, and looking at sort of what the tissues that are derived from those iPS lines over that time look like. So it's a generational study in people with multiple productions from it. So obviously it has not been done yet. But what I can say is that the majority of what is seen from making an iPS line, and we've made lines. There was a woman who actually was post-mortem who was 93, and they made an iPS line from her, made cardiomyocytes, and they looked, in sort of casual observation, were beating and were relatively normal. So the process of making an iPS cell does appear to sort of restart the biological clock. Whether that is will be backed up over time with more science is yet to be determined. Similar with the epigenetic question, it's just not enough time has gone by for people to look at these deeper questions about sublayers of what is going on within the epigenome to make sure that person X, Y, or Z that they're exposed to, it may relate to some functional observation that you can see in vitro. So those are questions that are beyond the scope of what is known today.
If you can sell to the Chinese, I bet you have the capital. Yeah. Because they are really environmental. It's on the top of their the 4th on the top of their 5-year plan. So, and they have 1 billion people.
Yeah.
Go ahead.
So I have a question for from the web that it's a little bit related from Todd Bredhauer, who I believe is at the Technical Support Working Group. And he asks, I think maybe you answered already the first part, but maybe not the second part. So he said, what is the prevalence of chromosomal anomalies to include epigenetic modification differences in tissue chip cells? And does this change as a function of time as the tissue chip approaches the end of its service life? I don't think you answered the second question already.
It's my lucky day today. So in general, iPS lines, the concept of a pluripotent stem cell is that it has the ability to proliferate forever. But every cell line, whether it's an ES line or whether it's an iPS line, ultimately will undergo some degree of chromosomal abnormality, and it can happen very early on in passages, or it can happen very late. And so there are still, from the ES work, a lot of Dr. Thompson's lines are still out there, H1, H9. They're heavily used, and the sort of standard of culture care is to make sure you look at karyotypic stability of your product, because it will shift with time, and it's something that we do with every line that we work with. We karyotype every 5 passages just to make sure that there's no chromosomal funny business going on. So it's a fundamental piece of the puzzle that has to be dealt with, and it's actually breakthroughs in that area would be very helpful, because it is labor intensive and expensive with today's standard of art for chromosomal aberration and detection. So that part is just part of the price of doing business at this point. And then the epigenetic one is similar to what we talked about before. It's a hot area of research, hot area of intrigue to understand. Is there a difference between an iPS line that you make from blood, from heart, from liver, from lung, and et cetera? And I think in general, the early work was, how do I say this nice, was probably clouded a bit by incomplete reprogramming. So taking a cell and reprogramming from one tissue wasn't completely transformed back and saying, well, this cell line that I started from that was heart, it makes heart better than neurons. A lot of that data now, I think, is that that line wasn't fully reprogrammed. And by definition of a reprogrammed line, it should be able to go down every lineage. And so to me, it's part of the rules of engagement of understanding what's the quality control for a reprogrammed line and making sure that those are equivalent when you're looking at one publication next to the other. But again, it's a hot area that is going to need to be well understood as this field moves from a research tool to a regenerative medicine therapeutic application.
Okay, Bill, thank you for waiting. You have the floor.
Bill Farland, Colorado State. I guess I'd like to take the comment that Jonathan made about the idea of taking known kidney toxicants and running them through the kidney test and flip that around a little bit and maybe direct this at Rusty. After I think a healthy skepticism about what kind of information we would get out of high throughput screens and in silico types of models, we've come away thinking that we do get some good information to work with. And a lot of that information could help us to choose chemicals wisely to do proof of concept on some of these various tissue platforms. And so I guess I'd like to hear from you your thoughts about that in terms of the ability to work with some of the developers, take the information from the high throughput systems and say, let's look for some good candidate chemicals to run through and do proof of concept.
So my personal opinion is one of the probably the most important lessons we've learned from the high throughput screening is that something we've intuitively probably knew from the beginning was the fact that many of the chemicals, at least the environmental and industrial chemicals that we're dealing with, act not like pharmaceuticals. So we kind of knew that probably going in, that they're fairly dirty, promiscuous chemicals, right? But one of the things that I think that the high throughput screening offers us is that we're now able to identify those chemicals which appear to be more selective in their interactions with the pathways or the biological targets. For the ones that are very promiscuous, I think that the tissue chips and that sort of technology isn't going to help us a whole lot. But I think for the ones that we identify that are fairly selective, we can use these more sophisticated culture models to sort of do what Rick was talking about as sort of a foundation to these AOPs, mode of action, right? Exploring how you get from a pathway perturbation to an ultimate apical response, a perturbation of apical response, and making that connection, right? And being able to be fairly confident that these mechanisms can be extrapolated to different susceptible populations and in different contexts, whether it be developmental context, age, different sexes, and so on and so forth. So I think that in many cases, it's those selective ones that these selective chemicals that these tissue models actually provide the most value to, at least in my view. Does that answer your question?
So we are at a break. Would you like to ask your question now or after the break? Okay, so what we're going to do is take a 15-minute break, and that would take us to 3:35. So thank you all for the participation. We'll come back and do this some more.